Hi everyone, this is Sungwon Lim, CEO and co-founder of a company called ImpriMed. What we are doing is we are providing an AI-driven functional precision medicine for cancer treatment. So today I'm gonna just talk about what that's the functional precision medicine means, and then especially the AI part. So I know this is an educational webinar, so I'm not gonna go too much details about our company's technology development. But I do like to start with some kind of basics about AI and how much AI is really how powerful the AI when it is combined with precision medicine, especially for the cell-based functional precision medicine. So I would like to start with a thank to the organizers of the Lab Roots who invited me to this talk, and I will start. So, cancer has been here with the humans for forever. It's been a long time. So that's why we always is trying to come up with a new drug development and a new technology development. But unfortunately, the cancer is one of the biggest the life-threatening disease for human. 26,000 people die of cancer every day. So one of our biggest question that started the ImpriMed was that how can we help cancer patients who need effective treatment immediately today? So we believe that I think many of the audience agrees with that. It's the key is the precision medicine. We can personalize what we currently have in our medical toolbox for individual patients to improve their treatment outcome. So precision medicine is not a new concept. It's been for a long time. So many researchers and clinicians have been working to approach this achieve the precision medicine concept. So people have been using the patient information and also drug sensitivity, immune subtyping, and the most popularly genetic information. So we know that the genetic information is find out the I know this is a genetics week, so we have been listening to a lot of great talks about genetics. Genetic information is amazingly important, and then we achieved a lot of things based on genetic information. But what I can see here is that when we just do all these kind of approaches individually, we saw some of the good outcomes, and then we saw some good achievements. But when it is approached individually, these rational approaches for precision medicine show the certain limitation, as you can see on the right side. So genetic information is definitely crucial, but what I can emphasize here is that alone is not sufficient to predict the cellular response to drugs in the patient body. So genetic information is definitely one of the most important origins of cancer development. But when it comes to the cellular information and then cancer cell information to the drugs and then inside the body in vivo clinical outcome, there is a lot of gap between the DNA mutation and then cells actual behavior inside the body. So what I want to emphasize here is that we want to combine the DNA information with the cell-based functional assays, because cellular information is also very important, what we call the functional information. And then this approach is called the functional precision medicine. So at ImpriMed, we combine this live cell analytics with artificial intelligence. What that means here is that we built the AI-driven prediction tool for oncologists based on the live cell analytics. So we receive the patient's fresh cancer cells and then fresh tissue sample biopsy from the hospitals. And then we have our centralized lab in California, Palo Alto, California, and Mountain View, California. And then basically what we are trying to do is that we are extracting as much information as possible from the live cancer cells from the patient. You can see that we are combining all different information with listed up in my previous slide: the patient information, drug sensitivity assay result, immune subtyping, and then genetic information all together. So if we collect all these raw data, it comes up to around more than 100 different features. How can we make this great, the most accurate prediction out of these 100 different features. It is very difficult using to digest by the human brain. So that's why we need the AI, especially the machine learning algorithm. So this machine learning algorithm is trained to digest all these input variables to spit out what is the prediction score or the prognosis evaluation for the in vivo drug response predictions. So this report goes back to the hospital within 7 days. So doctors can prescribe the individualized cancer treatment regimens. So by doing this approach, we can position between diagnosis and treatments. So after the patient is diagnosed, for 9 patients, right before the treatment, we can help the clinicians or oncologists to make the best decision. So this is a helper. We are the helping tool. We're providing a helping tool throughout this patient's treatment journey, not only the 9 patients, but also when it is relapsed. So we know that AI can do a lot of different things for cancer care. What I am focusing right now here is that we can use AI to do the risk screening, treatment response prediction, and the prognosis evaluation, so that we can actually streamline the finding out of the best drug upfront. So so by selecting the best drug for each patient upfront to improve patient's lifetime and then the quality of life, and then excluding the drugs that may not work for the patient, so we can reduce the necessary time and cost for that. So our goal here is that we're providing oncologists' best prediction tool for personalized cancer treatment. I have been emphasizing our live cell technology. So it is, I know many of you are very familiar with the lab cell lines, which is immortalized or just like, it does not die as long as you feed them with a sufficient nutrient. But it is completely different for the patient's primary cancer cells. So you can see here, this is lymphoma patient cells in a very nutrient-rich RPMI plus 10% FBS media, and then it just naturally die within 4 days. With this, in this situation, we cannot actually do this kind of actual drug sensitivity assay on it. So that's why we developed this ImpriMed optimal culture media that can maintain the cells alive almost 80% up to 4 to 7 days. You can see on the right side here is that the left column here is from the red line here without our optimal media, and then right column is with our optimal media. After 4 days, you can see the still intact lymphocyte population in our optimized media. So this is how we can save the cancer cells and then maintain the cells' original characteristics even after the sample biopsy. Let's go on to the AI. So I would like to start with the definition of the AI and then especially the machine learning. I know many of you are very familiar with AI and machine learning, especially these days, like ChatGPT and then all these kind of like OpenAI. So what is AI? So AI is actually, it's a higher level word of the machine learning. You can see here that AI here, you can see that a program that can sense, reason, act, and adopt. And the machine learning is included in the AI. It's part of the AI algorithms. Machine learning is a self-adaptive algorithm that can learn from data without using a complex set of rules. So what that means, I'd like to show an example of what this machine learning algorithm means. I'm going to show you some of my favorite the food photos. So can you tell what type of food this is? Yeah, yeah, definitely it's a taco. It's Mexican food. What about this? Yes, it's enchilada, Mexican food. So how were you able to tell this what type of this food is? Did you smell it or no, obviously not. Because of the cilantro and then all these kind of ingredients you can see, maybe. But what about this? You still can't tell this is Mexican food, that this is burrito, even though you cannot see all this ingredient inside. How are you able to tell this? Because you were trained to you were trained throughout all these kind of long years that you actually tasted it, and then you looked at it, and then you learned that, oh, these are the Mexican foods. So that's exactly what the machine learning does. So it's just algorithms to improve with all this kind of the exposed data. We can train the model, we can train the computer, this algorithm to spit out the best predictions, pretty accurate predictions. So with the new data, we can do the predictions. So after this training, so what machine learning algorithm can do is they are really good at complex tasks that require the analysis of large quantity of data. So they are really good at finding complex patterns and discovering unprecedented correlations, and then also identifying subtle biological differences beyond the human brain. So let's go into how can we use machine learning to improve cancer treatment. So we can use it for the disease diagnosis and then drug development, and also the treatment decision support, which I really want to focus throughout this presentation. So first, for disease diagnosis, I'd like to give you an example of a company called Lunit. Lunit is providing a machine learning-based imaging analysis software for early detection of cancer. So you can see here that this photo shows that Lunit algorithm, based on X-ray chest image of this old male patient, they actually detected this small subtle difference and a subtle difference in this X-ray image 3 years before being diagnosed. So definitely this Lunit AI prediction software can help radiologists to reduce their overall reading time and improve early stage lung cancer detection. What about for the drug development? So we all probably are, I assume that many of you guys are very familiar with the drug development process. So for the drug development, I actually have been working in the anti-cancer drug development for 10 years before I founded this company. So I'm very familiar with this space too. So for drug development, we are actually utilizing a lot of structure data, and then the chemical, genetic information, and also most importantly, clinical outcome data and then patient information. These are all the input for this the machine learning algorithm for the drug development. So we can use this algorithm so design the next generation of the new drug, and then also we can predict the patient clinical outcome based on digesting all these input variables. So we can use the machine learning for throughout all the drug development journey, starting from the designing, screening, manufacture, and also for the pharmacology, and then clinical trial, and then also for the drug repurposing. Then most important topic for this talk, how we can use the machine learning for treatment decision support. What that means is that we can actually use the machine learning algorithm to predict the patient clinical outcome, including response and then progression-free survival and overall survival. How we can do that? We can combine all this blue circle data from the hospitals, and then we can generate in-house the drug sensitivity, this red circles data, including drug sensitivity, immune subtype, genetics, and blood tests that I listed up, the number of data, and then the type of data that we're collecting at ImpriMed. AI is the bridge between this measured data or collected data and the clinical outcome, so that we can actually, this AI algorithm enables this personalized treatment. So how can we streamline the development of machine learning model for cancer precision medicine? Interestingly, our human's best friend, furry friends, our dogs are our key. So interestingly, every dog cancer patient is unique, just like similar to human. So at ImpriMed, we start to save our dogs first. So it is very sad to see that our dogs and cats, they live a shorter lifetime than humans. But what that means is that they actually have a very fast tumor progression, and then they show a 7 to 8x faster drug response. So we can actually quickly build and validate our AI models in these dogs and cat cancer patients very fast. And then even more, less regulatory pathways here, so we can actually collect a lot more data, a lot more sophisticated patient information from the hospitals. More interestingly, they actually get the same type of cancers as humans get, and then they are treated with the same drugs. So this means that once we collect our data and then build our technology here in the veterinary space, we can move on to the human side by help of these data and technology. So I told you that we can actually predict the response and then progression-free survival and then the overall survival. So how can we build this machine learning and use the machine learning for the response prediction? So I already showed you that our workflow, this loop, collect the sample and then run the assays and then provide this to the hospitals within 7 days with the prediction scores. So throughout this cycle, we are providing these information to the hospitals: drug sensitivity information, immunophenotyping, and then the most comprehensively, the evaluation, the prediction values, which drugs is best for this specific patient, and then which are predicted to be less effective. After 90 days, we collect this in vivo response data from hospitals. So we approach the hospitals again and then ask them, so what kind of drug did you actually end up with? And then what were the clinical outcome? Hospital has all this kind of data and record. They provide this the patient follow-up chart, so that we can actually compare our prediction and the actual clinical outcome data. So throughout this cycles, we can build this AI model for each individual drugs. The prediction score here is the output of the machine learning model in our single drug response model, especially for the dog lymphoma patients. So you can see here is that based on the prediction score, we can easily rank the drug in this order. So when veterinary oncologists to see this report, they can easily recognize that, oh, these top 3 drugs are better for this specific patient. And then, for example, I was planning to treat this patient with mitoxantrone, but I would not use it because it is predicted to be a low prediction score. What about the survival, especially for the progression-free survival? We can track the patients throughout like all the protocol, the combinatorial drug protocol, so that we can build this the progression-free survival machine learning model. So very similar to human hematopoietic cancers, especially for lymphoma and leukemia. Dog lymphoma patients, first-line chemotherapy is CHOP, C-H-O-P, the 4 different drug combinatorial regimen. Of course, in human, we combine the CHOP with rituximab, but the rituximab is antibody therapeutics, which does not work very well for dogs. So what we can build is that based on all this follow-up and all this collected data, we can do this risk stratification based on the progression-free survival. In terms of progression-free survival, where this patient is categorized into. So low risk for CHOP or high risk for CHOP. Low risk means that it will show longer progression-free survival. High risk means that it will show only a very short PFS for CHOP. So low risk, if a patient is categorized into low risk, that means that it's favorable for CHOP. We combine with this earlier response, what is the probability of CR achieving a complete remission in cycle 1 and cycle 2? So by combining these two together, we can provide this predictive information for the first-line chemotherapy, CHOP. So doctors can see this report and then they can actually interpret this report as, oh, this patient probably wants to go at least cycle 2, not just stopping at the cycle 1, going to cycle 2. And once it is achieved, it is predicted to show a longer progression-free survival. So we looked into all different type of lymphoma patients, even in the same immunophenotype, they show each patient show all different risk prediction, and then these are very like 80%, almost 80% correlated with the actual clinical outcome. That's the same for the T cells. So this is exactly why we need this personalized approach, personalized medicine approach for the cancer treatment. And what about the overall survival? In human side, we actually we are not only focusing on our dogs and cat cancers, but since 2 years ago, we started our translational research to the human side. So I'm gladly announced that our the human multiple myeloma machine learning paper has been accepted to the Nature NPJ Precision Oncology Journal 2 weeks ago. So what we are doing for this human multiple myeloma patient is that we can do similarly risk stratification, low risk and high risk, but for the overall survival instead of progression-free survival. So we built these two different regimen machine learning models for human multiple myeloma patients, especially naive patients who are not eligible for the autologous stem cell bone marrow transplant. In this way, we can actually categorize the patient into these 4 categories, and so that doctors can easily see that, oh, for these patients, RD regimen will be better than the VMP regimen, and it actually the clinical outcome show the significant difference for these patients. So all these approaches, I'd like to show how we are actually using this machine learning algorithms in the real world patients. So Ellie was 11.5 years female terrier mix, unfortunately diagnosed with high grade lymphoma. And then we received the patient sample from the Seattle Blue Pearl Veterinary Hospital from Dr. Kevin Choi. Based on, so we looked into the Ellie's medical record written by Dr. Choi. He said that if this patient is a B-cell lymphoma, I would like to treat this patient with a drug called Tanovea. Tanovea is one of the 5 drugs that are approved only for dogs, dog cancers. But when we actually looked into our immunophenotyping, it turns out that it's not a B-cell lymphoma, it's a T-cell lymphoma. Moreover, Tanovea was ranked as a weak drug in our prediction report. So, but we are not the decision maker. We provide this predictive information to the hospitals, and the doctors decide, usually for the veterinary space, in discussion with the pet owner. So we were wondering that which drug was actually selected. Doctors selected the chlorambucil, which was the second best drug, not the Tanovea. They excluded Tanovea. And the reason to select the second best is that the chlorambucil is almost 1/10 less expensive than the meclorethamine, and then it's a orally prescribed drug, but meclorethamine was an IV drug, so it's easy to administer and then it's less expensive. So it was great to hear that doctors actually followed our prediction report. But more important question here is that, so how did Ellie respond? So Dr. Choi sent us this photo with his email after several months later. So with a graduation cap, so Ellie completed all this chemotherapy cycles. And then this is the actual quote from the Dr. Kevin Choi. So without our prediction report, he would not select this chlorambucil because throughout his 12 years of his practice, he did not see this drug was very effective. So, but he trusted our platform, and then it was how this was how he was able to extend Ellie's life 6 more months in a very healthy condition in complete remission. He actually, without our service, Ellie was expected to live a little longer than 1 month. So Ellie actually lived 8 more months after the ImpriMed aided chemotherapy, which corresponds to 5 years of human age. So we have so far we provide our service to 4,000 dog blood cancer patients, and then we are seeing a lot of great results here, especially for Yoki and Samson. They live more than 3 more years, especially for Yoki. Yoki went through, for the last 3.5 years, Yoki relapsed 4 times, and then every time we came in, and then we provide the new prediction reports for this evolved Yoki's evolved the tumors. So, and then we were every time we were correct. Doctors follow the top 2 or 3 drugs treatment regimen, and then it extended 3.5 years of Yoki's lifetime, which corresponds to more than 20 years in human side. So we have a lot of like the publications. So I will just go through not too much detail, but our first paper shows that we can actually reduce time to achieve clinical remission if doctors use the higher score drug. And then I already showed you that for the CHOP patients, we extend the disease-free, the progression-free survival before relapse. And this was the last year's presentation at the Veterinary Cancer Society meeting. We actually looked into the dog with relapsed B-cell lymphoma. Based on the top 3 drugs, doctors who followed 2 or 3 drugs in our top 3, the prediction scores, they showed almost double the overall response rates. And that was actually showed similar in the T-cell lymphoma, which is more hard to treat compared to the B-cell lymphoma patients. So for T-cell lymphoma patients, our machine learning model separate out the the doctors who actually follow this high concordance group is who followed our AI prediction results. So they shows a lot more overall survival, not only the progression-free survival, but overall survival compared to the low concordance group. And then also there we show the huge difference in the clinical remission response. So we can show that patients who were actually given the treatment in high concordance with ImpriMed predictions, they show the better response, and then they live the longer. So can we also help future patients? So what I showed you here, providing this predictions service, the precision medicine service to the current patients, we can improve their patient lifetime and then the quality of life. But can we also help future patients by improving drug development process? Yes, we can do, because of this live cell, the functional precision medicine technology. So this ImpriMed live cell technology is very different from the lab cell line or animal model cells. It's actual patients, fresh live cells. So what we can provide to the pharmaceutical companies that we can test this new anti-cancer drug compound the pharmaceutical company developed, and then we can come in before the clinical trial to provide them that which of the final 3 or 4 candidates show the best cell cytotoxicity on patients, their target patients' live cancer cells. And then we can also provide the responder versus non-responder profile. And then we can also identify the synergistic combination with the conventional drugs. So before the clinical trial, we provide this information to the pharmaceutical companies so that pharma companies can design a better clinical trial to improve the clinical trial success rate, so they can refine the inclusion criteria. And then during the clinical trial, we can come in and then we can work with the pharma companies and then also the clinical trial CR company to provide this live cell analytics throughout this clinical trial, so that we can enable the pharma companies be equipped with the companion diagnostics when they get approval from the FDA. So not only for the pharma companies, but we can also are open to all this work with the researchers in this field. So especially FDA earlier this year, President Biden signed this FDA Act 2.0, which no longer requires animal tests before the human clinical trial. So this platform can help a lot of patients, the future patients. So this live cell plus AI platform, not only for the current patients to improve their treatment outcome, but for the future patient, we can increase the clinical trial success rate for novel therapeutics. So I would like to end up this presentation with how we can develop this successful machine learning in precision medicine. just like similar to the other engineering process, we have to design, build, and test to learn. We have to make this cycle like again and again. To do that, we need people, technology, material. People means data scientists who are equipped with knowledge in biology and in clinical science and in data science. These are the people who are specialized, different from the IT data scientists. And then technology. Fortunately, these days there are a lot of publicized, published AI algorithms that we can use start with. These basic algorithms start with this and then refine those algorithms. But most importantly, we need to set the direction based on the clinical needs and then start with the right algorithm for that purpose. So we always need to talk to the clinicians and then listen to what our oncologists and then clinicians talk about. And then material. So identifying the right or proper input features for this machine learning is very important. And then collecting the real world clinical outcome data is very important. So I'd like to emphasize this data collection. This is what we are generating at ImpriMed. So drug sensitivity assay, testing all different drugs on the patient live cancer cells. Based on the drug sensitivity alone, our prediction accuracy, our machine learning algorithms prediction accuracy was about like 60%. When we add the immune subtype, it increased a little bit. When we add the blood test data and then patient information, you can see the plus PI, it actually improved our the prediction accuracy significantly. Then we are adding the genetic information here. So we recently found out the novel biomarker that shows a significant difference in the clinical outcome. So altogether, this is very important. So collecting all these features, right features, is very important. And definitely, collecting the clinical outcome data is one of the most important things to develop this machine learning model. This is the clinical outcome data for training and validation. These are the ground truths. So we have to train our AI algorithms, the machine learning algorithms, using this clinical outcome data. So I told you that one of the biggest benefit with working with this veterinary space is that we can collect this clinical outcome data a lot quicker and then a lot more comprehensively. So far, we were able to collect the 2,500 clinical outcome data who were actually treated with the drugs. They are naturally occurring patients who were actually treated in the clinics. So this is one of the greatest reasons that we are working with our dog and cat cancer patients, and then we are moving on to save their pet parents, which are humans, next. So I would like to end up with this acknowledgements. These are the ImpriMed team. I would like to thank our co-founder and then also our bioscience team, data science team, software engineering team, business and commercial operations team, and then our advisors and consultants. This is the last slide. I'd like to share the quote from my own blog posting, the journal paper on this innovative veterinary care journal. So AI cannot replace the clinicians or the cancer biologists. So AI is a valuable tool that can help clinicians do their jobs with greater speed and precision, freeing them up to focus on essential workflow and concentrate on patient welfare. So AI is a great tool to make your life easier, and then just focusing on the more important thing, and then improve your productivity, and then you can actually focus on the more important thing, the patient welfare. So this is my email address here. So if you have any questions, please contact me. I will do my best to answer all the questions. Thank you.